Welcome back to the deep dive. This is where we take those dense stacks of sources, the foundational research, the key articles, your notes, and turn them into the essential knowledge you actually need.
And today, we're really jumping into the deep end.
We're tackling a field that, honestly, it still sounds like something out of a sci-fi movie, but it's already here. It's already the engine behind some of the biggest life-saving breakthroughs we've seen.
Nanotechnology in medicine.
Exactly.
And it's such a non-negotiable deep dive for anyone tracking progress, really.
Yeah.
For so long, the word nano was, it was just for theoretical physics or movie plots.
Yeah.
But like you said, this isn't fiction.
Not anymore.
I mean, the tech that delivered those incredibly fragile mRNA molecules for the COVID vaccines to billions of people.
Yeah.
That was a marvel of nanomedicine. It really is the defining technological shift for 21st century health.
That sets the stage perfectly. So our source material today is this incredible collection of foundational modules on the topic.
Yeah.
And our mission for you is to pull out 3 core insights. First, we have to get the physics. Why does shrinking something down to 1/1000th of a meter just completely change how it behaves? Second, the engineering. How did nature get there first? And, what tools have we built?
And third, the big one.
The big one, the ethical and logistical checkpoints. As we move from these carpet bombing treatments to true targeted precision medicine.
And you can't even start that conversation without first just grappling with the scale,
Yeah.
the sheer mind-bending scale of it all. We have to start with the nanometer itself.
Right.
Because if you don't get that dimension, none of the rest of it makes any sense.
Okay, so let's try to unpack this. A nanometer, it's 10 to the minus 9 meters, 1/1000th of a meter. We throw that number around, but it's so abstract, it's almost meaningless, right?
It is. The sources use this powers of 10 perspective, which I think really helps it click.
Give it to us.
Okay, so imagine you take a regular marble, something you can hold in your hand. Now, shrink that marble down in your mind until it's just 1 nanometer wide.
Okay, impossibly small.
Right. Now, at that scale, if your marble is 1 nanometer, the entire planet Earth would only be 1 meter wide.
Wow. Okay, that forces a mental reset.
It does. A single strand of your hair, which feels so thin, is still 80 to 100,000 nanometers wide. We are operating at the level of DNA, of single proteins.
And this idea isn't new, is it? The concept has been around for a while.
The seed was planted way back in 1959. The physicist Richard Feynman gave this legendary speech. There's plenty of room at the bottom. He basically challenged the entire scientific community. He said there's no law of physics that stops us from manipulating matter, atom by atom.
He laid down the challenge, but the tools just weren't there. How do you build something that can see, let alone move, an atom?
Exactly. The breakthrough, the real physical breakthrough, didn't come until 1981, the invention of the scanning tunneling microscope, or STM.
And that changed everything.
Everything. For the first time, scientists could actually see individual atoms on a surface. And even more critically, they could pick them up and move them. You could literally write with atoms. That moment, when room at the bottom became a real, tangible engineering feat, that was the ignition switch for the whole nano era.
So we've got the size, 1/1000th of a meter. But like you said, size alone isn't the magic. What is it about this specific range, the 1 to 100 nanometer range, that makes it this golden range?
It's that transition zone. That's the key. Below 1 nanometer, you're basically just dealing with individual atoms, small molecules. That's pure chemistry.
Okay.
And if you go above 100 nanometers, the material, it just starts acting like bulk matter again. It follows the rules of the world we see every day.
And in our world, the big rule is gravity.
Gravity rules. But when you shrink down into that golden range, something else takes over completely.
And that is?
Surface forces. The first huge rule change is the dominance of surface area. Think about it. If you take a big object and smash it into tiny pieces, the total volume, the amount of stuff, is the same. But the amount of outside the exposed surface area just explodes.
The sugar cube rule from the sources is the best way to picture this.
It's the perfect analogy. You drop a sugar cube in your iced tea, and it dissolves, slowly, because only the atoms on the outside of the cube can touch the water.
But if you crush that same sugar cube into a powder,
it dissolves instantly.
Yeah.
Because you've massively increased the surface area. The takeaway from medicine is profound. Reactions happen on the surface. Nanomaterials have this incredibly high surface area to volume ratio, which makes them chemically hyperactive. They can be powerful catalysts, or they can make a stubborn drug dissolve instantly. That's the first big advantage.
Okay, so that's surface area. But then things get even weirder. We move from classical physics to quantum mechanics, quantum confinement.
This is where it gets really interesting. because the laws of physics themselves actually change for electrons at this scale. In a big chunk of gold, for example, the electrons can move around freely, like swimmers in an ocean. They have a continuous range of energy levels.
But when you shrink that gold down to a tiny nanoparticle,
Now those electrons are trapped in what the source calls a tiny room. And because their movement is so restricted, the rules of the game change. They can't have just any energy level anymore. They're forced into specific, quantized energy levels. It's a direct consequence of their confinement.
and this quantum shift means that just by changing the size of a particle by a few nanometers, you can completely change its fundamental properties.
Completely. Let's look at the property shifts. First, color.
Right. Bulk gold is yellow. We all know that.
But nanogold, suspended in a liquid, is a vivid red, or sometimes purple. That color change has nothing to do with chemistry. It's purely a function of its size. The quantum confinement changes how the electrons interact with light.
It's not just color, though. Strength is another one.
Look at carbon. We know it as soft, flaky graphite in our pencils. But when you arrange those same carbon atoms into a flawless nanoscale tube, a carbon nanotube, it becomes 100 times stronger than steel at a sixth of the weight. We use them in high-end sports gear for that reason.
And even melting point. Bulk gold melts at, what, over 1,000 degrees Celsius?
164 degrees Celsius.
Mhm.
But a tiny 2.5 nanometer gold particle, it melts at around 600 degrees. The atoms on the surface are just less constrained. Their bonds are weaker.
So the ability to tune all these properties, color, strength, conductivity, just by tweaking the size, that's the core of the whole thing.
That's the power. And medically, this translates directly to things like solubility. You can take a drug that's oily and won't dissolve in our water-based blood, shrink it down, and that massive surface area forces it to dissolve, making it more available to the body.
It's incredible that we can engineer things at this scale, but we have to give credit where it's due. Nature was the first, and still the best, nanotechnologist.
Oh, absolutely. Evolution has spent billions of years perfecting these structures. It's a huge source of inspiration. We call it biomimicry.
Let's start with the classic example, the lotus leaf. The way water just beads up and rolls right off, taking all the dirt with it. That self-cleaning property.
The secret isn't some super smooth wax. It's actually the opposite. The surface of the leaf is covered in these tiny, perfectly arranged nanobumps. These bumps trap a layer of air between the water droplet and the leaf.
So the water is barely even touching the surface.
Exactly. It's floating on a cushion of air. The contact area is minimized, so the water's own surface tension makes it ball up and roll away, cleaning the leaf. We've copied that exact nano-architecture for hydrophobic sprays you can buy for your clothes.
Okay, and then there's the gecko. The gecko's ability to just walk up a glass wall, that's pure nanoscale physics.
It's gravity-defying and leaves no residue. A gecko's foot is covered in millions of tiny hairs called sedae. But then each of those hairs branches out into millions more, even tinier nanohairs.
So it's hairs on hairs on hairs.
Down to the nanoscale. And these nanohairs get so incredibly close to the surface that they leverage something called van der Waals forces.
Which are those super weak molecular attractions that are usually totally negligible in our big world.
They're negligible one-on-one. But the gecko is creating billions of these contact points simultaneously. It's the cumulative strength of all those weak forces added together. And we've copied this too. There's something called gecko tape now, a dry adhesive that works on the same principle.
So we learned from nature's blueprints, and then we built our own toolkit. The sources break down the nanomaterials for medicine into 4 main classes. Let's start with the soft ones, the organics.
Right. These are made from the building blocks of life lipids, proteins. They're valuable because our bodies generally recognize them, they're biocompatible, and they can be designed to safely biodegrade when their job is done. The star of this class is definitely the liposome.
The famous hollow fat bubble.
That's a great way to put it. It's a sphere made of a double layer of fatty molecules, just like our own cell membranes. This structure is incredibly versatile. You can hide a water-soluble drug in its hollow, watery core, or you can embed an oil-soluble drug inside the fatty wall itself.
And this is the tech from the COVID vaccines.
This is exactly what protected the mRNA. It was a liposome bubble, shielding that fragile genetic code from being instantly destroyed by enzymes in our body, long enough to deliver its message to our cells.
Okay, so from soft materials to hard materials, the inorganics.
These are your metals, your metal oxides, things like gold, silver, iron oxide. Unlike the organics, they're generally not biodegradable, but they offer incredible stability and unique optical or magnetic properties you just can't get otherwise.
Like the gold nanoparticles we mentioned.
Exactly. Their red color makes them perfect for diagnostics. They give a very clear, easy-to-read signal. And in cancer therapy, we can use lasers to heat them up. They convert light to heat so efficiently that they can literally cook a tumor from the inside out with incredible precision.
Third class, the carbon-based materials, the superstars, like carbon nanotubes.
Their structural perfection is just unmatched. Sheets of carbon rolled into these flawless tubes. In medicine, we see them used a lot for biosensing, making ultrasensitive electrodes, or in advanced medical composites where you need something incredibly strong but also lightweight.
And finally, you combine them, the hybrid nanomaterials.
This is where you get the best of both worlds. You might take an inorganic core, say magnetic iron oxide, and then wrap it in a biocompatible organic liposome shell.
And what would be the point of that?
The key example is a magnetic liposome. The organic shell lets it travel safely through the body, but the magnetic core gives you a superpower. A doctor can use a strong external magnet, completely non-invasively, to physically pull and concentrate those drug-filled particles directly to a deep-seated tumor.
That's incredible. You're literally steering the drug through the body.
It's the exact spot you need it. It maximizes the dose where it counts and minimizes it everywhere else.
Which brings us to the fundamental why of all this. Nanomedicine really exists to fix the huge built-in problems of conventional drugs. The term the sources use is carpet bombing.
It's a perfect, if a bit grim, analogy. When you swallow a pill or get a standard IV, that drug goes everywhere. It floods your entire system just to reach one tiny target. And that leads to three big problems.
First is toxicity, and chemotherapy is the textbook example.
Right. Chemo drugs are designed to kill rapidly dividing cells, which is great for cancer. But our bodies are full of healthy, rapidly dividing cells, hair follicles, the lining of your stomach, the bone marrow cells that make your blood.
And the drug can't tell the difference.
You can't. So you get the devastating side effects: hair loss, nausea, a weakened immune system. That's a direct result of poor targeting.
Problem number 2 is solubility. So many of our best drugs are essentially like oil.
They're hydrophobic, and our blood is mostly water. So these drugs just don't want to dissolve. They clump up. To get around this, drug makers have to use much higher, sometimes dangerous doses just to get a tiny fraction of it to work.
And then there's the biggest hurdle of all, what the sources call the liver tax.
The first pass metabolism.
Yeah.
When you swallow a pill, its first stop after the gut is the liver. And the liver's job is detoxification. It sees this drug as a foreign chemical, and its mandate is to destroy it.
So it's doing its job too well.
Far too well. It's not uncommon for the liver to wipe out 90% of an oral drug dose before it ever even gets into your main bloodstream to do any good. We're losing almost all of it right at the start.
Okay, so that's the carpet bomb. How does nanotechnology switch us to the GPS-guided missile?
It offers three fixes at once. First, target delivery. We can coat the outside of a nanoparticle with molecular keys called ligands. These keys are designed to fit only the specific locks or receptors found on diseased cells.
So a key for a breast cancer cell won't fit a healthy lung cell.
Exactly. The payload only goes where it's supposed to. Second, controlled release. The nanoparticle acts like a tiny time release vault. Instead of one big spike of drug in your system, you get a steady therapeutic level for days or even weeks.
And you can even make that release intelligent, right?
You can. You can design the particle shell to dissolve only when it senses the specific environment of a disease. For example, tumors are often slightly more acidic than healthy tissue. So the particle stays intact everywhere else, but as soon as it hits that acid, it breaks open and releases the drug.
And finally, beating the liver tax. How do you do that?
This is so elegant. We can engineer the nanoparticles so that instead of being absorbed into the blood from the gut, they're picked up by the lymphatic system.
The lymphatic system.
Think of it as a secret tunnel. The lymphatic system completely bypasses the liver on its first pass. By taking this route, the drug stays 100% intact, meaning we can use much smaller, safer, and more effective doses.
So even with a perfectly designed particle, the body itself is a fortress. It has all these barriers. The first one is the cell membrane itself, right? The particle has to get inside the target cell.
Right. You can't just diffuse through. It has to be actively brought in. The cell has a process for this called endocytosis. It literally envelops and swallows the particle.
And how it swallows depends on the particle.
It does. The cell has different loading docks. For big foreign things, it uses phagocytosis, which is like cell eating. But for our smaller therapeutic nanoparticles, it might use clathrin-mediated endopsychosis, where it forms a little protein-coated pit to bring the particle in.
Okay, but then there's the ultimate security gate, the blood-brain barrier, the BBB.
It is truly formidable. It's designed to protect the brain, and it blocks something like 98% of all potential drugs from ever getting in. The cells that line the brain's capillaries are sealed together with these incredibly tight junctions. There are no gaps.
Which is why treating brain tumors is so difficult.
Notoriously difficult.
Yeah.
So to get past it with nanotech, we use what's called the Trojan horse trick.
Okay, I'm intrigued.
We leverage the brain's own needs. We coat the nanoparticle with a ligand that mimics something the brain needs, like transferrin, which is used to transport iron.
Ah, so the barrier's security guards see the particle not as a threat, but as a delivery of essential supplies.
Precisely. The receptors on the barrier see the transferrin, grab onto it, and actively shuttle the entire nanoparticle across the barrier and into the brain tissue. It's a way of tricking the body's own transport system.
That is genuinely brilliant. But, okay, let's say the particle gets past these physical barriers. The moment it hits the bloodstream, it faces another challenge: the protein corona.
This is maybe the single most important factor that decides a nanoparticle's fate in the body. The second a particle hits your blood, proteins start sticking to it. Thousands of them. They form this layer, this corona, all over the surface.
So the body's immune system never even sees the nanoparticle we designed. It only sees this coat of proteins.
That's it. And that protein coat becomes the particle's biological identity. If the wrong proteins stick, what we call a hard corona, the immune system's trash collectors, the macrophages, see it as an invader and destroy it immediately.
The sources use the Roman effect analogy for this.
It's a great way to think about it. It's like a celebrity walking into a party. At first, all the small, fast-moving proteins rush in and stick to the particle. But over time, they get pushed off by bigger, more specialized proteins that have a stronger attraction. It's that final, stable layer of proteins that tells the body, friend or foe.
So, is there a way to control this, to make the particle invisible?
There is. This is the whole reason for something called pegylation. We attach these long, flexible polymer chains called polyethylene glycol, or PEG, to the particle surface.
What do they do?
These PEG chains act like molecular whiskers. They create this slippery, water-loving shield around the particle that physically prevents most proteins from being able to stick to it. It essentially gives the nanoparticle a stealth coating, making it invisible to the immune system and letting it circulate for much, much longer.
Okay, so that's stealth. Let's talk targeting. We have two main strategies. The first is passive targeting, which sounds less precise.
It's less precise, but it's incredibly effective because it exploits a fundamental flaw in tumors. It's called the enhanced permeability and retention effect, the EPR effect.
Well, leaky pipe analogy.
Leaky pipe. Tumors grow so fast they need tons of new blood vessels.
Yeah.
And they build them sloppily. The vessels are disorganized, full of holes and gaps. They leak.
Unlike healthy blood vessels, which are tightly sealed.
Exactly. So nanoparticles, if they're the right size, around 100 nanometers, can slip right through those leaky gaps and into the tumor. And here's the retention part. Tumors also have terrible lymphatic drainage. So once the particles leak in, they get trapped. They accumulate there naturally.
And this effect is the whole basis for one of the most successful nanomedicines ever, Doxil.
Doxil was the first one approved by the FDA, and it's a perfect case study. It takes a powerful chemo drug, doxorubicin, which is known to be very damaging to the heart.
Right, cardiotoxicity.
And it seals that drug inside a pegylated liposome.
So it's got the stealth coating?
It has the stealth coating, and it's sized perfectly at about 100 nanometers. So it's too big to leak out of the tight, healthy blood vessels around the heart. But it's the perfect size to leak through the sloppy, gapped vessels in the tumor. The result: the drug builds up in the cancer and leaves the heart alone. A huge reduction in side effects.
That's passive targeting. But for true precision, we need active targeting, the real lock and key.
Right. Active targeting is when you take that stealthy, pegylated particle and you deliberately decorate its surface with those molecular keys we talked about. A great example is using the vitamin folate.
Why folate?
Because many types of cancer cells, like ovarian or breast cancer, are metabolic furnaces. They're hungry. And they have way more receptors for nutrients like folate on their surface than healthy cells do.
So you're baiting the hook.
You're baiting the hook. The cancer cell sees the folate-coated particle, thinks it's a vitamin it desperately needs, and actively swallows it. It invites the Trojan horse inside. This massively increases the amount of drug that gets right inside the sick cell.
The sources also detail other kinds of carriers. We've talked about liposomes. What about dendrimers? They're described as being tree-like.
They are fascinating. They're synthetic molecules built up layer by layer from a central core with these perfectly branching structures. Their advantage is total architectural control.
And what does that control get you?
It means you can attach multiple things at once with incredible precision. You can hide a drug in the core. You can stick a targeting key on one branch. And you can attach an imaging dye, a little glowing tag, to another branch. One single dendrimer becomes a multi-tool for therapy, targeting, and diagnostics all at once.
And for those really tough, oily drugs, there's a simpler solution, nanomicelles.
The metals are brilliant because they self-assemble. They're made of molecules that have a water-loving head and an oil-loving tail. When you put them in water, they automatically form a sphere with all the tails tucked inside, creating a tiny oily pocket in the middle.
A perfect trap for an oily drug.
A perfect self-forming trap. It's a simple and effective way to make an insoluble drug soluble in the bloodstream.
Finally, let's circle back to controlled release. We have the matrix-based model and the diffusion-based model.
Right. The matrix model is like a dissolving sponge. The drug is mixed evenly throughout a biodegradable polymer. As that polymer sponge slowly dissolves over days or weeks, the drug is steadily released.
And the diffusion model is the tea bag.
The tea bag. This is the more tunable one. The drug is concentrated in a central core, and it's surrounded by a semi-permeable membrane. The drug then slowly diffuses or seeps out through tiny pores in that membrane. And scientists can control the size of those pores to regulate the release rate with incredible precision.
Okay, let's shift from treatment to detection. Nanodiagnostics. This feels like a huge paradigm shift. We're not waiting for symptoms anymore.
It's moving healthcare from reactive to proactive. The goal is to find disease at the molecular level. Like the analogy says, not waiting for the fire, but sensing the first molecule of smoke.
And these smoke molecules, these biomarkers, are incredibly rare in the blood, right?
They're like needles in a haystack. Too faint for most traditional tests to pick up. But this is where the massive surface area of nanoparticles comes back into play. We can coat a single nanoparticle with thousands of antibodies, which act like tiny magnets for that one specific biomarker. It massively amplifies our ability to find it.
Which improves both sensitivity and specificity.
Exactly. Sensitivity to find the tiniest trace, and specificity to make sure you're only grabbing the thing you're looking for.
So let's talk about the tools. Nanobiosensors. The most common ones are optical, right? They use light.
Yes, and the perfect example is the rapid COVID-19 antigen test we've all used. That little red line is pure nanotechnology at work.
So explain the magic. What's happening there?
It's a phenomenon called plasmonic resonance. The test strip has gold nanoparticles that are coated with antibodies for the virus. If the virus is in your sample, it acts like a bridge, causing the gold nanoparticles to all clump together at the test line.
And that clumping is what we see.
When the gold particles are spread out, they don't really have a color. But when they clump together, the way their surface electrons interact with light changes dramatically. And that change causes them to absorb light differently and appear as that strong, vivid red line.
That's amazing. Okay, but what if your sample is opaque, like blood? You can't see a red line, so you use magnetic sensors.
Right. For those, we use superparamagnetic iron oxide nanoparticles, or Aspirants. Same principle. Coat them with antibodies, they find their target, they clump together. But instead of a color change, the clumping changes the local magnetic field, which is very easy for a sensor to detect no matter how murky the sample is.
Nanoparticles are also changing medical imaging, acting as better contrast agents. For CT scans, we can use gold.
Gold is way better at absorbing X-rays than the traditional iodine contrast dye. So if you can get gold nanoparticles to accumulate just in a tumor, it will light up on the CT scan with incredible sharpness, giving a surgeon a perfect map.
And for MRIs, it's iron oxide again.
Yes, the superparamagnetic particles. In an MRI, they disrupt the magnetic field in a predictable way that creates negative contrast. So tumors or disease spots in the liver show up as very dark areas against a light background. And a bonus is that our body can naturally process and clear the iron over time.
Okay, now we have to talk about quantum dots. These seem like they're from the future. Tiny crystals whose color depends only on their size.
It's a pure quantum effect. You can have a quantum dot made of, say, cadmium selenide. If it's 2 nanometers big, it glows bright blue. Make that exact same material 5 nanometers big, and it glows deep red. You get a whole rainbow of colors from one material just by controlling the size.
Which allows for something called multiplexing.
Yes, tracking many things at once. Imagine a surgeon injects a mix of quantum dots. The blue ones stick to the tumor. The green ones stick to the major blood vessels. The red ones stick to nerve bundles. Now, in a single real-time image, the surgeon can see the tumor, the blood supply they need to manage, and the nerves they absolutely must avoid.
There's a catch here, though, isn't there? A lot of these quantum dots are made of heavy metals like cadmium. That sounds like a long-term safety problem.
It's the major bottleneck. That bioaccumulation risk is very real. A huge amount of research right now is focused on making safe-by-design quantum dots out of things like carbon or silicon to get the same amazing optical properties without the heavy metal toxicity.
Let's pivot to one last, really urgent application. Fighting antibiotic-resistant superbugs.
This is a global health crisis, and nanomedicine offers a completely new angle of attack. Traditional antibiotics usually target just one specific thing in a bacterium, and the bacteria eventually evolve a way around it.
They learn the trick.
They learn the trick. Nanoparticles, on the other hand, attack in multiple ways at once, a triple threat.
Let's start with silver nanoparticles. They're in wound dressings, right?
They are. Silver releases ions that are toxic to bacteria. They get inside and bind to the bacteria's DNA, essentially gluing it shut so it can't replicate. It's incredibly effective against things like MRSA.
And then there's copper nanoparticles.
Copper is great for contact killing. On hospital surfaces, for example. When a bacterium touches copper, the copper ions trigger a massive, overwhelming burst of reactive oxygen species, ROS.
A chemical store inside the cell.
A chemical firestorm. It punches holes in the cell membrane, shreds proteins, destroys its energy centers. It's a multi-front attack that happens in minutes.
So that's why resistance is so hard to develop. The nanoparticle attacks with the physical breach, the ROS chemical storm, and DNA disruption.
A bacterium might be able to evolve a defense against one of those, but evolving a simultaneous defense against all three is almost impossible. It's a huge advantage in the fight against resistance.
Okay, so far we've mostly talked about passive carriers, but the real frontier, the sci-fi stuff, is nanorobots. Active programmable machines, swallowing the surgeon.
This is the ultimate goal. But the engineering challenges are just immense. You're talking about a machine, 100 nanometers or less, that needs its own power source, propulsion system, sensors, and logic.
How are researchers even approaching propulsion? How do you make something that small move?
One promising idea is a catalytic drive. For instance, you could coat one side of a nanobot with platinum. Our bodies have hydrogen peroxide in them naturally. When the platinum touches it, it creates a chemical reaction that shoots out tiny oxygen bubbles, pushing the nanobot forward like a tiny jet.
So you could guide a nanodriller to a blocked artery.
That's exactly one of the goals. It could use enzymes or physical vibration to gently break up plaque from the inside, no surgery required. Or you could do precision nanodissection, using a tiny laser to cut a single filament inside a rogue cell to stop it from spreading.
The complexity here, designing and guiding billions of these things, it's staggering. This is where AI has to come in.
AI is the non-negotiable partner. It's the mind, and the nanoparticles are the hands. For diagnostics, for example, nanosensors create a flood of data that no human could ever process. Only a deep learning algorithm can sift through that and spot the 4-cell cancer cluster that a pathologist would miss.
And for treatment, there's the concept of the digital twin.
The digital twin is revolutionary. AI builds a perfect virtual replica of your specific tumor based on your genetics and imaging data. Then it can test thousands of different nanodrug combinations on your virtual tumor to find the absolute optimal treatment before you ever get a single dose.
AI is even changing how we invent these materials in the first place.
It is. It's a field called materials informatics. Instead of spending years in a lab with trial and error, an AI can simulate millions of hypothetical nanoparticle designs in a few hours and predict which ones will be the safest and most effective. It massively accelerates discovery.
Which brings us to the crucial counterpoint, safety, nanotoxicology. The very things that make these particles so powerful also make them potentially dangerous.
The number one long-term risk is bioaccumulation. Our bodies haven't evolved to get rid of some of these synthetic materials like carbon nanotubes or heavy metal cores, so they can get stuck. They build up in the liver, the spleen, the bone marrow, and they can stay there for years.
And that chronic buildup can lead to genotoxicity.
Damage to our DNA. Over time, these trapped particles can generate those ROS chemical storms, causing chronic inflammation and potentially damaging the DNA in our cells, which could lead to cancer or other diseases decades later. Managing this long-term risk is critical.
And what about the environmental impact, the Trojan horse effect?
Right. When antimicrobial silver and copper nanoparticles from our clothes or water filters wash into the environment, they can kill the good bacteria needed for sewage treatment. Worse, they can stick to other pollutants like pesticides and carry them up the food chain, concentrating the poison.
This leads right into the big ethical questions, the slippery slope between therapy and enhancement. If a nanobot can fix a damaged heart, should we use it to build a super heart that never gets tired?
The line is getting so blurry. Using nanotech to restore sight is therapy. Using it to give someone infrared vision is enhancement. And if these enhancements are incredibly expensive, you risk creating a two-tiered society, a biological divide between the haves and the have-nots.
And then there's data privacy. If these nanosensors are collecting real-time data from inside my body, who owns my biological secrets?
It creates a massive power imbalance. Could your health insurance company raise your rates because a sensor detected a pre-symptomatic marker for a disease you won't get for 20 years? Could an employer decide not to hire you based on your internal stress level data? The potential for misuse is huge.
So how are regulators handling this? The rules are still being written, aren't they?
They are. The FDA in the US takes a product-focused approach. They recognize a nanoparticle isn't just a chemical, so they require extra data on its physical properties, the exact size, shape, surface charge, because a tiny change in any of those can completely change how it behaves in the body.
And in Europe, they're focused on the precautionary principle and a concept called nanosimilars.
This is a huge challenge. A generic version of a nanomedicine isn't just about getting the chemical formula right, it's a physical object. If the generic version has a slightly different size distribution or a different peg coating, its safety and effectiveness could be completely different from the original. Regulating that consistency is incredibly difficult.
We have covered so much ground here. We started with the fundamental physics, that golden 1 to 100 nanometer range where surface area and quantum confinement just change all the rules.
Then we saw how we can move from the toxic carpet bombing of old drugs to the precision of a nanomedicine like Doxil, which uses the EPR effect, the tumor's own leaky architecture against it.
And we looked at how this same tech is creating a diagnostic revolution, sensing disease at the molecular level with the potential for theranostics, the ability to see and treat a disease all at the same time.
And we wrapped up by seeing that AI is essential to designing and guiding this future, but that all of this power comes with a heavy burden of responsibility for safety, ethics, and privacy.
So, after all of this, what does it all mean? The big challenge of this nano age seems to be about permanence. We learned that the greatest long-term risk is bioaccumulation, these non-biodegradable particles getting stuck in our bodies forever with unknown consequences.
And if we're getting good enough to design materials to be permanent and ultra-effective, our ultimate responsibility isn't just in making them, it's in making sure they can be safely removed or dissolved once their mission is complete.
Which leaves us with a final provocative thought.
We're entering an era where materials must be designed not just for function, but for their own flawless, non-toxic disposal. So the question you need to mull over is this. If the risk lies in permanence, in irreversibility, how aggressively must we commit to safe by design principles, ensuring that every single thing we put into the body also has a reliable, non-toxic self-destruct switch built right into its very core?
A profound thought, shifting the focus from invention to responsibility. Thank you for joining us for this essential deep dive. We'll see you next time.